Mine. Joe worked on the role of hormones and song learning in zebra finches when he was in my lab, but rapidly moved on to bigger and better things. Since Joe graduated, he and I had a number of conversations in which we discovered we have something in common in why we chose science. And that is because I went to a Bolivian Catholic convent school as a child, and Joe went to a similarly tight religious school in high school. And both of us found ourselves questioning some of the dogma we were learning and wondering what the evidence was and what the data was. And so we are both very data driven. And that fact, plus the fact that Joe is the most senior of the people we're hearing from today. So we are going to be hearing data from Joe.
Thank you, Heather, for that generous introduction. Thank you, Heather, for that intro, and thank you also to Christine for organizing logistics for this event. I graduated from Williams in 2009, so it's great to be back here, and I'm excited to talk with you all. So today I'm going to focus mainly on my current work as a research scientist at MIT, but then at the end I'll talk briefly about the path I took from Williams to where I am now. So first we'll talk about my current research, which is focused on the development of dendritic inhibition in the mouse visual cortex. In the developing cortex, each neuron receives many excitatory and inhibitory inputs. And this balance of excitation and inhibition is critical for healthy development. Here in this drawing, I represented excitatory pyramidal neurons in magenta and inhibitory neurons in the cortex. Most neurons are excitatory, and most of what we know about the development of the cortex comes from excitatory neurons, and particularly excitatory synapses on excitatory neurons. So from studies of excitatory synapses, some key themes have emerged. The first is that early in development, there's a period of exuberant synapse formation when the number of synapses increases dramatically. And that's followed by a period of pruning when the number of synapses decreases. The second theme from excitatory synapses is that their development is experienced. So for example, mice are born with their eyes closed, and then when they're around 12 or 13 days old, they open their eyes. And excitatory synapses mature in response to visual experience after that. There's also a critical period later in development when neurons in visual cortex are sensitive to changes in visual experience. So these themes come from excitatory synapses. What about inhibitory synapses? By comparison, we know far less about the development. One thing we do know is that there are multiple inhibitory cell types that innervate pyramidal neurons. So here's our excitatory pyramidal neuron array. We have somatostatin expressed in inhibitory neurons, which target primarily pyramidal dendrites. A somatostatin is a neuropeptide released by these neurons, and the expression of somatostatin defines its capacity to target dendrites. We also have multiple non-somatostatin types that target the area around the pyramidal cell line. Now, this is an oversimplification. There are many different inhibitory subtypes, but for today, we're going to focus on somatostatin versus non-somatostatin subtypes. Most inhibitory work on the development of inhibition focuses on synapses that are on or near the cell line. And part of the reason for that is it's much easier to detect inputs if they're on near the cell line. It's much more difficult to detect inputs that are on the cell line. Because of this, we know far less of the development of inhibition of dendrites, even though the vast majority of synapses are on dendrites. So this is the focus of my work. Today, we're going to talk about the development of dendritic inhibition, particularly from somatostatin expression. And from here on out, on the slides, I'll introduce somatostatin. So if you want to study somatostatin innervation, you way to visualize it. So as I mentioned on the previous slide, it's difficult to detect these inputs. See the vision. Particularly want to see boutons, which are the structures that house presynaptic sites, because those presynaptic sites are for that information. To map that boutons, we use a genetic strategy that crossing two boutons with each other, so that the offspring will express the fluorescent label in their somatic pattern boutons. So the outcome here is fluorescently tagging boutons, which are here represented in cyan of our somatic pattern. This is what those boutons look like. This is a coronal section from a mouse's brain. We have our somatostatin boutons in cyan, and we have dapping, which stains all nuclei and fluorescent various primary visual cortex. So I have a full set of primary visual cortex from the surface of the brain all the way down to the corpus. If we zoom in on this small rectangle, you can see individual somatostatin boutons. So when I talk about bouton density on the next slide, so this is ask, when is somatostatin boutons development? So here on the y-axis, we have somatostatin bouton density. Go from left to right, right. Example, just ranging from and all the way through. And we're imaging visual cortex. So the relevant milestones here are eye, which happens around 12 or 13 days old in mice, and a critical period when neurons and visual cortex are sensitive to change. When we look at the trajectory of bouton density over time, what we see is that at postnatal age 10 5, there's still very diffuse somatostatin. And then in the days after IUD, during this result is interesting because it suggests that there could be a causal relationship between IUD and this metastatic. I want to acknowledge two other members who worked with me on this. Danielle Helene was an undergrad, and she worked with me on this. She's student. Kendall Bernal was she worked with me on this. If you remember the introduction, I talked about a theme from the development of excitatory synapses where there's a period of pruning when their numbers decrease during adolescence. What we found for somatostatin is there's no period of pruning. So once they initially are development, they're maintained. So this result suggests that there's a developmental shift in the balance of adaptation dendrites, because we know that dendrites are excitatory at a time. So far, what I've shown you are boutons, and we're assuming that those boutons represent synapses. Here in this by drawing, we have our bouton in cyan, and I'm representing presynaptic protein in red, postsynaptic protein in yellow, and our postsynaptic proteins. But so far, I haven't shown you any synaptic proteins. So can we confirm these boutons have synapses? Now, it's difficult to visualize synapses. They're small and they're very close together. So this is just one synapse, this drawing. But in reality, the brain is more like this. And there's an axon together. We're going to see all the synapses. And it's difficult to assign a synapse to visual bouton because there are nearby boutons that synapse belong to. So how can we get back to a view like this where we see individual synapses? So to resolve individual synapses, we use a technique called magnified analysis of proteome or MAP. A MAP involves physically expanding and clearing the tissue. So this is a mouse brain before expansion and clearing. This is the same mouse brain after expansion and clearing. So by physically expanding the tissue, we spread the synapses farther apart. So we can effectively zoom in on an individual synapse instead of having a crowd of massive synapses put together. To look for synapses, we do immunohistochemistry for synaptic proteins and then imaging on a standard confocal microscope. So with this technique, we're effectively achieving super resolution imaging by scanning the tissue, even though we're imaging on a conventional microscope. This is what our map images look like. So in cyan here, we have our semester one enabled random dendrite. If we zoom in on this small square, here's the dendrite. So this is the dendritic shaft. Here's the spine neck. So with the tissue expanded, we really appreciate how big the spine neck. So the scale bar here is 1 micron, which in the expanded tissue, which corresponds to about 250 nanometers of original dimensions. In cyan here is a somatostatin bouton. Now we look at both pre and post-synaptic proteins. So for our pre-synaptic protein, we're using this, which is found in both inhibitory and excitatory pre-synaptic sites. For our post-synaptic protein, we're looking at which is specific to inhibitory post-synaptic sites. So On this individual somatostatin glucan, we see two distinct synapses, each of which has its pre and post-synaptic protein. So with this technique, we can confirm whether our somatostatin glucans are forming synapses. I worked with another postdoc in the lab on this portion of the project. So with this technique, we asked, do somatostatin glucans have synapses, especially early in development? So here's an example of glucan at post-synaptic. Here's another example of glucan at post-synaptic. Now in this P18 glucan, we have one synapse in our post-synaptic. But in this P18 example, we have no synapses on it. So when we quantified the number of synapses per glucan, we found that, to our surprise, when mice were 10 days old, more than a third of somatostatin glucans lacked synapse. Now, this was surprising to us because we were using glucans as a structural surrogate for synapses. And that is valid later in development. So even by posting the the vast majority of glucans have at least one synapse. So that group having zero synapses are small. But early in development, many glucans still lack a synapse. So P10 to 18 is clearly an important time for synapse formation on somatostatin glucans. But what else is happening at P10 to 18? Mice are opening their eyes experience. So because synapse formation and the onset of visual experience are happening at the same time, yes, does somatostatin synapse formation depend on visual experience? Now, to ask this question, we dark reared mice, meaning we place their cages in a dark room and we use negative goggles to use. For the sake of time, I'm not going to go through each of the experiments. We did 3 different experiments with dark rearing, but the conclusion from all of them is that neither somatostatin on more synapse formation depend on experience. So regardless of the age of the sample or what parameter we measured, we found no effect of dark rearing. This was surprising to us because we know that a different type of does require like also excited for somatostatin around be different cell types. So to summarize what I've shown you so far, we saw that somatostatin glucan and synapse formation lack efficient bias. We also saw that unlike excitatory information, somatostatin information is not processed and also does not depend on visual experience. Now these results have implications for how we understand certain information, because we see that somatostatin neurons follow a different developmental trajectory from excitatory neurons, even though those excitatory neurons are the main postsynaptic partners. And our results also suggest that there's a shift in the balance of excitation and inhibition on dendrites during ages when dendrites are losing their excitatory intent but maintaining perfect. So far, what I've shown you are static snapshots of synapses and fixed tissue. What's happening in the live brain? We follow synapses over time. Are they dynamic? Are they lost? Even if they're net as being stable. So to track synapses over time in live mice, we do a cranial window surgery, where we remove a portion of the mouse's skull and we replace that portion of the skull with glass. So here in this photo, we're looking through the glass into the brain of a live mouse. Now, what are we imaging through these windows? We have fluorescently labeled pyramidal neurons here drawn here in magenta. So here in the drawing, we're zooming in on the dendrite. So we have our inhibitory postsynaptic sites along the dendrite in yellow, and then we have our somatostatin glucan site. So here we have both pre and postsynaptic structures that we're imaging in vivo as the mice are alive. And because the mice are alive, we follow the same neurons over time as mice are growing up. So here's an example of neuron that I imaged from the time the mouse was 22 days old, barely weaning age, all the way through 42 days old, which is mid-adolescence. Now, when we're imaging these neurons, we have a view of the full arc. And the dendritic arc is a complex 3D structure. So to appreciate the structure of the arc, let's get a video of this same. So now we're looking at a pyramidal neuron in the brain of a live mouse. This is a side view, so the top right now is the surface of the brain. Now we're going to rotate. When we finish rotating, we're going to be looking down at the cell from above. So now we're looking down from the surface of the brain, looking into the brain of the cell. The cell is in there 2-3, it's a pyramidal neuron. We have our dendrites in magenta and our somatostatin glutons in cyan. So as we zoom in, you can start to appreciate individual somatostatin glutons. And now I'm going to tilt this field of view slightly so that you can appreciate there are multiple dendrites passing over each other in this 3D field of view. So next, I'm going to hide the gluton channel So this is a three channel image. I can hide the on so that we can focus on the dendrites. So here along the dendrites, we have our inhibitory postsynaptic sites yellow. So each of the dendrites has its own resonant inhibitory postsynaptic sites. And as we zoom in, we can appreciate that there are inhibitory postsynaptic sites along the dendritic shaft. And there are also inhibitory postsynaptic sites on some of the spines. For example, here's the spine that has a postsynaptic site. So that was a 3D view. Now let's zoom in on two individual synapses from the same neuron. So here's the full dendritic armor. And we're going to zoom in on just this very short segment of that, right? So here we have our dendrite, our inhibitory postsynaptic sites are here, and our somatostatin. So here's an example of an inhibitory postsynaptic site that has full spatial overlap with the somatostatin neuron. So this is an example of a putative somatostatin synapse because we're seeing both the pre and postsynaptic structure associated with this synapse. This inhibitory postsynaptic site, on the other hand, has no spatial overlap with any somatostatin neuron. So this is a putative non-somatostatin synapse. So we've only fluorescently labeled the somatostatin neurons. There are other inhibitory cell sites whose glucons are not fluorescently labeled. So when we don't see a fluorescent glucon, such as here, we assume that that inhibitory input must come from a non-somatostatin cell site. Now, the reason we care about presynaptic cell type is that different inhibitory cell types have different primary patterns. So this postsynaptic neuron will experience that input differently depending on which inhibitory cell type applies. But this mouse is alive, so we can follow the same dendrite over time. And we can look at the same synapses later in development. So here, in the image we just looked at, the mouse was 22 days old. And here, 12 days later, the mouse is 34 days. So when the mouse is 34 days old, we see those same two synapses resolve later. Now we see a new synapse in the spine. So the spine itself was there earlier. Now that spine is synapse. So from the data we analyzed so far, we see that inhibitory synapses have a high rate of loss during development, even if they're dead as but we can also ask more nuanced questions. For example, are synapses more likely to be gained and lost depending on whether they contact a somatostatin neuron or another organization or cell type device? We can also ask whether synapses are more likely to be gained or lost depending on where they're located on the arc. Now, the reason we care about location on the arc is that the influence of the inhibitory will differ depending on where it's located. For example, it's far from the cell body, or very close to the cell body, or if it's on the shaft, like synapses, or on the spine. So the analysis for this portion of our project is still in progress, so stay tuned in the future results. For not only thank the talented lab, especially those who work directly with me on this project, who I can call throughout the presentation. The rest of the lab being such great colleagues, and I do have one more slide. So my biology research experience here at Williams, I worked with Joan and studying plant pollinator interactions about more relationship. So that's that. Then in some places, general. So I want to talk a little about this step in my career because it's what I did right after. Part of the reason I chose to do this was right. So I was going through a lot of find my point in my whole life. I'm so glad that I took 2 years to work instead of jumping straight into press. So this was a fantastic research experience. In terms of research skills, I learned to do training degrees during that time in general. And I got excited about the people. I got excited about grad school where I was clinical work, looking at the role of banana hormones in habits. During my PhD thesis work, I used both imaging and video. Now, my postdoc, as we discussed, I continued my interest in animals brain development. Now it's time about so in my future, I hope to have my own lab where I can combine the genetic labeling strategies I use now with the which I use grad school study animals and development of brain structures. So thank you so much for your attention. Happy to answer questions about either the research or the path of the career path.
That was really cool. Um, was the science captured in similar ways?
Yeah, so that comes from, so that summary graph was adapted from a review article. There are many types of data. So there's EM data, which is often not cell type specific, but can study the overall number of excitatory synapses. And then most of that data comes from spine imaging of different cortical areas. And the spine imaging data shows that that pruning is cell type specific. So that graph that I showed applies very well to layer 5 pyramidal neurons and their equal dendrites, shallow layers, but does not necessarily apply to layer 2-3 pyramidal. But it is consistent across cortical regions from frontal cortex to sensory cortices and in a different species as well.
Then a quick follow-up. So you left us with the cliffhanger, why it aligns with the eye opening any years.
Yeah, this is a topic that I'm currently speculating about as I write a discussion section. So one possibility is that dendritic inhibition is really important for the computational capacity of the dendritic arbor for compartmentalization. So it may be that a certain amount of dendritic inhibition needs to be in place to enable the computations that the dendritic arbor has to do in order to learn, especially, I mean, for mice, these ages are when they're reaching independence for the first time. When they start having visual experiences, also they're about to enter the world where they have a lot of risks. So they need a certain amount of dendritic inhibition to enable the experience-dependent plasticity that's going to happen in that stage of their life. So whether that's true across different sensory areas or across species would be interesting to know. But in terms of the critical period timing, there is some evidence that somatostatin neurons might regulate critical period plasticity. So in adulthood, manipulating their activity can reopen critical period plasticity, and transplanting embryonic somatostatin neurons can also reopen critical period plasticity. So historically, PP neurons have been thought to be responsible for regulating critical period opening, but it could be the somatostatin neurons.
Follow-up question.
I don't think so because they have no visible language. So that one aspect of this could be activity driven, which actually I can said as well, it's question as well. And retinal waves will persist whether we know the endogenous development proceeds. So my speculation is that even if I retinal waves will
in the experience that you had just after Williams going to MGH, I feel like it's hard to like dive right into a PhD program. I'm an off-cycle student and it would be weird sort of finishing Williams and trying to apply to things and then doing programs after that. How was your experience? And I figured how long were you at Tech? And
yeah, 2 years. Yeah.
And do you think that like really convinced you to do a PhD? Did you already know you were going to do a PhD? You wanted to take that time to like are you?
Yes, I knew I wanted to do PhD, and I consider applying straight out of plans. Reasons that Grace also discussed that application process. And I was job, had my honors thesis class schedule, and so and I was transitioning, so I had a lot going on personally as well. That I thought I just I need that time. Because I had done a technique in Windows that when I was interviewing for grad school, was appealing to lab setups.
Can you talk a little bit about the transition from MGH, looking at Alzheimer's and more neurodegenerative diseases to like moving towards neural development? So really, they're very nuanced differences, and I'm curious as to the transition from like both sides.
Yeah, that's a really good question. So here is an anomaly in terms of my research interest, and in some ways, grad school and postdoc, I returned to these interests, which were critical periods of plasticity and also role of hormones and regulating plasticity, just something I hope to return to in the future. If I find that. So yeah, when I was looking for research assistant jobs, I didn't know what research topic I was on, so I cast pretty wide net, and I interviewed at various places, and this seemed like a very I guess supportive and collaborative research environment where people were working together and friendly and teaching each other, and it seemed like I would learn a lot in that environment, and I'm glad I chose it, even though Alzheimer's, but I continued the technical skills that I learned were really valuable. And I also had the opportunity to present at the Society for Neuroscience conference while I was here, and what struck me about this lab was that technicians were able to have more intellectual ownership of projects than at many other places. So that was another thing that drew me to that lab versus the other ones I interviewed at.
There's one more sciencey, but are the somatostatin neuron positive imaging necessarily cortical neurons?
So this is an excellent question, and we have not explored that directly. We do know that there are cortical somatostatin neurons that have long-range projections, so they could be from there's 6 neurons from other regions here. Most of them we expect to be from local neurons, but it is very much fully explored. And with the approach we're using, where we have the glutons rather than the reference cell label, it would be difficult to trace where they're coming from. But with other tracing strategies, like what you use, explore that.
Anything else? Well, I want to thank our speakers very much. All of you.